Brush, and I'm a professor in geography and environmental engineering in the engineering school. I've been here for quite a long time, and I teach courses in ecology and in plant geography. But the research I do mainly has to do with collecting sediment cores from the Chesapeake Bay to understand the history of what has happened to these to the ecosystem. We take these cores, like you see here, and this one, for example, that sediment may have been deposited over a couple of thousand years. When the colonists came here, it was around the end of the 1600s, the earliest colonial settlement, though there were Native Americans, but there were not very many of them here, so they had very little effect. But once the colonists came, what they did very quickly was to transfer this whole forested system to agricultural land, and that happened over a period of less than 100 years, and the whole eastern part of the country was deforested almost completely, from 90% forest to less than 10%. So by the beginning of the 1900s, this whole landscape was pretty much deforested. What has happened as a result of that is a lot of the sediment and nutrients and fertilizers from the land were deposited in the estuary through runoff, and that has created conditions of eutrophication and anoxia, which means that these organisms cannot live in the estuary. They die out. The large fish, the oyster industry went down, and so on. So the question that came up in the 1970s, early 80s was, well, what happened to the bay? Why did this deterioration occur, the loss of the economy? I had suggested that one way to find out would be to take these sediment cores and look at the history. So that's how we began to do that. So we get this core, and then we cut it into sections, 1 centimeter sections, and then we have to date it. And it's dated by carbon-14. We send out this bottom samples to a lab that measures carbon-14. And we get a date. So the date with carbon-14 can vary from 500 years to 50,000 years, sort of. So it's important to date each sample of the core in order to get a kind of a chronology. So you can take a sample and say, okay, this sample represents, say, 1800 to 1850 or something like that. So once we do that, then we extract from the core chemicals like nitrogen, some metals, carbon, phosphorus, and so on. And we measure that in micrograms per square meter per year. So you have a sample of core, and we send these samples out to laboratories that do these measurements. And so we know in this particular sample we have so much nitrogen. Or we look at the molar ratios between the different nitrogen compounds. And that gives us an idea of the amount of nitrogen at a given time. And then we can look at another layer. And so we get these profiles then which show us the really huge changes that have occurred at a particular time. Since we have the core dated, we know when those changes occurred. And then we can go back to the historical records and see what was going on at that time. And there are very detailed historical records with from the agricultural census, which occurred since 1840, and then other historical records which indicate just what was going on the land. In fact, some of the records are the amount of fertilizers that were purchased per county So the other thing that's very interesting that we've learned from these cores is that how different areas are depending on the geology and the substrate. So we have taken cores from the Patuxent River, which is on the western side of the Chesapeake, and also from the Nanticoke, which is on the eastern side of the Chesapeake. The Patuxent is surrounded by soils that have a lot of clay and silt in them, and the Nanticoke by gravels, pretty much gravels. And when we look at the metals in those cores, they're consistently about, I think, around 10 times higher in the Patuxent, where the soil is a silt and clay where these metals would attach to those particles, than in the Nanticoke, where you have gravel and the metals are just going to run through that, get deposited somewhere else. So one of the things that our work is showing is how important that geology soil substrate is, which was formed over 1 million years ago in this area, and everything else like climate and land use is sort of superimposed on that template, so that you will. So that's why you have why the metals show up differently on one substrate than on another. So you have to be sort of aware of the effects of fertilization and of land use and of climate too are going to be different. They may occur synchronously, but they're going to be different at one place or another, depending on what that substrate is. And the other thing is that none of this really was understood until we did the paleo work, because you don't have that history. You know, what are you going to compare things to? Why did submerged macrophytes die out? Well, some people were talking about populations boom and bust, but that was not the case. You know, when we looked at the history, that most recent demise of these plants occurred only that one time, at least over the last few thousand years. So the research is important in understanding actually the economy of the Chesapeake, which was a huge fishery, an oyster industry, which died out completely. So economically, that resource is gone. And so we were able to show why the resource is gone. So before we began doing the sediment core work, there were all kinds of reasons proposed for this. One was overfishing, which is important too. What we were able to show is that it was really the runoff of nutrients, chemicals from the land that really was responsible for the demise of this ecosystem and the economy that went with it.